Hello and welcome. You're logged into the 8th of 12 Berry Soil Nutrient Management webinar sponsored by the Northeast Sustainable Research and Education Program, Northeast SARE, and hosted by Cornell University Department of Horticulture and Cornell Cooperative Extension. My name is Kathy Heidenreich, and I'm going to be your moderator at least temporarily as Jeff Miller from Oneida County Cooperative Extension is having audio issues. If he gets those straightened out, he'll be back with us shortly. We're going to be starting in just a minute. As before we begin, I want to remind everyone that you can put any comments or questions in the chat box. I want to let you know that Bob has uploaded a file to the file box, Soil Health Testing and Adaptive Management 2010. It's a paper that he wrote. He'll be telling us more about that later. I want to remind you about the web links. Northeast Share is there in the bottom right, and the webinar archive. So if you've missed any of the previous webinars, you'll be able to go to that link and listen to those. Remember that you can shift to full screen during the presentation if you'd prefer not to be distracted by what's going on maybe over on the right. If you hit the full screen button, you'll see just the presentation. And then again, if you want to have some other input, the little fellow on the top of the screen with his hand in the air, there's several options there for you to make some sort of a comment through that. Okay, I hope everyone is in and warm and safe. It's pretty snowy here. There's a pretty serious weather warning out there, so drive carefully on the way home. And with that, I'll introduce our speaker for today. Our speaker is Mr. Robert Schindelbeck, better known as Bob, and Bob is an Extension Associate in the Department of Crop and Soil Science at Cornell University. He has responsibilities in research, teaching, and extension. Bob coordinates the Cornell Soil Health Lab operations and has been a part of the Cornell Soil Health team's development of soil management strategies to address laboratory-measured soil constraints. He's produced numerous peer-reviewed and extension articles and has delivered over 200 presentations related to tillage, soil compaction, and soil quality at farmers' twilight meetings and professional conferences. Bob is going to talk to us today about soil health assessment. Thanks, Bob. Over to you.
Thanks, Kathy. Well, hello, everyone. I'm looking forward to this opportunity to share with you some information about the Cornell Soil Health Test and how it could apply to berries and how it does apply to berries. I should start out with some very basic introductory ideas because I think they're very useful in the whole context of developing testing, soil health testing in particular, in a new field like berries. There isn't a formalized soil health test yet for berries. There could be something that's very specific to berries, and I think in the course of this presentation, what I'll try to do is bring up issues and points that would allow kind of a shift in the focus of the test away from vegetables, which is what it was designed for, more specifically to berries. It doesn't necessarily limit the utility of the test. It's just that it could be ever better for berries. Okay, so we started the test. We actually began offering this test back in 2007 to the general public. We've done thousands of samples on both research and commercial farms in New York and throughout the entire country. We get samples from all over, including Canada, and we've learned an awful lot about soil health testing. And so I think this is a good opportunity to, as I say, disseminate this into the berry realm. I'm also going to be building on some of the ideas that Harold Van Ness presented back in, I guess it was October of 2011 in this to the berry group called Introduction to Soil Management and Berry Production. And I want to acknowledge too, first of all, we always talk about soil health, our soil health group as a team. I think it's a very important concept to keep in mind that as a group of interested, concerned scientists and educators and growers and field staff, there's so much we can bring to bear if we can have a common a common goal, and that's sure what happened here. I've listed some folks that were on our core team from its inception back in the early 2000s. Harold Van Ness, of course, is I work directly with Harold in CSS. Bianca Mobius-Clune, John E. Dow Wu, he's now at New Mexico State University. We're the crop and soils folks, and then George Bowie and his then graduate student Beth Gugino in plant pathology brought a lot of information to bear, and Dave Wolf over in horticulture, a real soil biology expert. We all kind of got together and kind of shared a common goal to kind of understand some of these soil health concepts. But again, without the growers and extension educators and field staff to kind of focus us and keep us on track, sometimes it's easy to get lost in minutia. But we just had a really good run of a really good team of people where we could really iron out some nice ideas, and a lot of important funding. The Northeast Region SARE, we got 2 large grants from them, 03 to 06 and 06 to 09, which is really where we were able to do the foundational work for this support from the Northern New York ADP. We have a research farm up at Willsboro where a lot of beta testing went on. The New York Farm Viability Institute helped us tremendously with funding support, even with a subsidy for New York growers to participate in the soil health testing. Of course, IPM helped fund a long-term research up at Geneva under George Bowie. And yes, I guess that's where I'll stop with all the introduction. Okay, so what is soil health? I think Harold might have shown this slide. It's so important to realize that when we talk about something like health and its capacity to function, these are very qualitative terms, or soil quality. They're qualitative. Yet as scientists, we want to measure things. We want to be quantitative. So it's kind of it's a bit of a challenge to measure a quality. So I'll show how we go about that here. Okay, and so with a healthy soil, I would say they're somewhat easy to even spot at a distance. The crop looks good, soil looks good. We kind of know what a good soil, again, qualitatively looks like. And I've listed some ideas here, some important features of a healthy soil. It's got good tilth. Again, it's very qualitative. Sufficient nutrients, depth, it stores water, it's disease organisms, good populations of beneficial organisms, and really at bottom, resistance to being degraded. Let's see if I can grab my arrow here. A resistance to being degraded and a resilience. A quick recovery from adverse events. We see a lot of extreme weather anymore, and your better soils, at a glance, the healthier soils, they just rebound faster, and so the plants are less stressed. Conversely, general signs of poor soil health, we can kind of see that something's going on. We would say something's going on here. Now, what exactly is it? First, we'd list the kind of these qualitative features. They often come up claddy. We'd say the soils are very hard. Sometimes there's a lot of runoff. The water doesn't seem to go in the soil. Plants don't germinate well. General declining of yields. So what we really want to do then is find out more specifically what is going on in the soil. What is actually not functioning. Another thing to keep in mind while we do that, as scientists, we're reductionists, so we want to put a very sharp point on every idea. Yet these ideas still have to exist in an interactive world and in an ecology. So we want to keep this in mind, that as we deconstruct and go way down into specifics, we never want to lose sight of the fact that these specifics operate in a holistic ecology. This complexity of interaction between these different, if you will, arenas of soil physicality, chemical, and soil biology is really part of what I call nature's beauty. These things just it's all interdependent. So I'll talk a little about that now. So here's an example of this interaction, and Harold had talked about this as well, but we see a hard soil with reduced rooting. So I've kind of highlighted in blue here some of these physical um, uh, factors to kind of talk about a compacted dense layer. Restricts our rooting volume. So then the plant can't extend its roots into a large volume of soil. So if there's an extended drought, there's less water to draw upon and less nutrients. But at the same time, this compacted soil causes a reduction in beneficial biological processes because of lack of aeration. So we see how there's this kind of it's not necessarily a straightforward, simple effect of what does hard soil do and hard soil reducing rooting. What I'm trying to suggest here is all these concepts kind of go in under this hard soil reducing rooting heading, but the idea is we want to keep all these ideas in mind at simultaneously. So we want our RAM filled with these sort of accessory ideas that kind of support this concept, because we'll need that to understand just what's going on then for understanding the soil testing, as well as then ultimately the soil management. Okay, so let's get to some soil chemistry. Some of you may recognize Carol McNeil. She's a vegetable crop specialist in many counties in central New York, standing here in front of a grower field that's looks like it has some issues. This field actually tested very high in nutrients, but as you can see, the stand's very poor. The plants are really suffering. And what we see here is, if you notice right in front of Carol, some key issues that one thing that our soils investigation showed us was that the soil was used very intensively by the farmer, and over time we saw that the organic matter was decreasing, the soil was getting tighter and denser, water wasn't getting in, and the soil was becoming quote unquote sick. You had it like a syndrome. You use it, you till it, you loosen it, but then it quickly gets back in this condition. And we, of course, intuitively know that there's more going on here than a lack of soil fertility per se, but just exactly what is it? And this is the sort of thing that really interested us at the beginning on our soils team, to kind of get at just what is going on here. We need more information in our soil testing. Here I show how we use soil nutrient soil chemical testing and nutrient recommendations. I show a strawberry test here taken from some Molly Shaw's work, where we can test the soil in something like a strawberry. We can even do foliar testing for these high value crops. As well as on the right here, I show a picture of a soil report where we get this is a modified Morgan extraction, where we get the actual levels of these different nutrients in the soil, where they low, medium, high rankings, and then even a recommendation on what to add. So we've come a long way in soil nutrient testing and recommendations over the last, really since World War II. So back to this ecology here. I want to come back to this. It's really a philosophy of recognizing that we want to go beyond just in the upper right here, the soil chemical testing, but realize that it's really a three-legged stool. We've got soil, there's a physical environment and a biological environment that we need to have. All these need to be sound, or using our three-legged stool analogy, the stool can tip over based on one of those being weak. So that this kind of gets to part one of the talk today, and that's we want to measure different aspects of the soil, so we can identify constrained soil indicators. Then part two is then what are we going to do about it? So we're going to use this tear-apart reductionist strategy to go into each of these soil arenas one at a time, but always keeping in mind that we're going to use that same information later as we put this information back together towards dealing with our soil and managing it in the best way we can. So here we go. This is I'm going to start with the soil chemical testing here. Um, what we see here is the um, right in soil chemical testing, very important issue of pH. pH controls the solubility of a lot of metals and just a lot of nutrients in general. And so the soil is really a chemical repository of nutrients. But moving down to the biological arena, we also know that the soil is also a repository of life, and this cycle of life, for example, decomposing organic matter, releases a lot of chemical compounds. And in this cycling, there's also a lot of pest suppression. So again, kind of suggesting this kind of interdependency, so that we aren't just growing in clean sand and we're adding the appropriate chemicals. We have a living, breathing biological environment here that's also affecting these chemical levels and responding to them. And then finally, moving to the physical arena, I've heard the soil physical structure called the house, where all these where the roots live and where all the soil biological processes take place. Certainly, what's listed here kind of makes sense. We need a place where water and where the plant can grow, where water can come in and be stored, also where water can exchange with air. And so the soil can breathe and get fresh oxygen and remove carbon dioxide. And then it's also a place, the soil is also a place where all these processes take place. So if our house is badly damaged, then our biological processes, which may release chemicals to the plant or make chemicals more or less available, will be affected. So I want to stress again, we really can't talk about any one of these without talking about all of them. But again, we're going to take this reductionist approach first to deconstruct it, but keeping in mind that we want to put this all back together. Okay, I've shown here some photos taken from our laboratory. These are the tests that we perform in the lab. And I'll talk more about these in a minute. I just wanted to kind of show a picture so you get an idea. These are fairly straightforward tests, nothing super involved. Well, I'll go through them one at a time later. But across the top, I show the soil physical tests. Then across the bottom, the soil biological analyses. And of course, the soil nutrient testing is mentioned in the middle. We use a modified Morgan extraction to then score the soil nutrient levels. So I'll go through these now in detail. Okay, let's start with this idea of the soil house. So a soil aggregate. I'm a soil physicist. I'm very interested in soil aggregates, soil crumbs. What you see here are very small particles of soil, kind of all joined together, glued together, if you will, into a large crumb or an aggregate. There's large pores and small pores. Again, as a soil physicist, I'm probably more interested in the void spaces and what's in them than the particles themselves. These different pore sizes perform different functions. The large pores, of course, rapid water intake and drainage, and then air intake. The smaller pores, of course, store water over time. As these smaller pores stay full of water, this is where the soil organisms would live, in that water. So this is just a model of the soil house. On the left here, we see a good soil structure where we have well aggregated soil that's infiltrating water rapidly between the particles. We always want to take in rainwater that comes. On the right, we see as these aggregates break down, it's actually a surface compaction. The soil closes up. I'll use my arrow here. I think I can. I had trouble with it before. There we go. As these aggregates then break down, they end up self-clogging, and we end up with it's really a surface compaction, and then the water can't get in and exchange. As the water moves in, of course, air moves out, stale air, then fresh air can move in behind the water. So we want to definitely protect these aggregates so that we can have this sort of model function here on the left. And keep in mind also that these large pores are the most sensitive and most likely to be destroyed by compactive events. Here's a couple pictures from the field of what we mean by soil structural breakdown. I'm sure we've all seen this. In particular interest, I think, is on the bottom left here. It's a winery in California trying some different management strategies to between these semi-permanent rows of grapes to try to increase their water intake. You see on the I guess the left side of that picture how the water is just kind of standing at the surface, kind of puddling up there. Whereas in the kind of the middle of the slide, they've tried some other things that kind of have the soil loose, and the water seems to be infiltrating better. I wanted to show this slide, this picture, in contrast to, in particular, the pictures just above it, which were taken from just a more of a vegetable field, a potato field actually, where we'll get to this later, but keep in mind that when we discuss these topics, we're going to also have to discuss where they're occurring. Across this whole field at the top, we would sample and test usually the entire field. We don't necessarily focus our quest for information. Whereas in the winery example, you can see how right next to the plant, where would be a very important place to sample in for example, a cornfield. We aren't really even managing in this picture, managing that area in the same way. So it's we want to keep in mind that these processes occur everywhere in a vegetable field, but more localized in some of the berry or specialty vegetable areas. Okay, so that actually our way of testing this the soil structural stability in the lab is we use a rainfall simulation, really. We apply drops, a half-inch of raindrops in the course of 5 minutes onto some soil crumbs. And we can see on the left versus right here the sort of result we get. At the bottom, it shows where these soils came from. This is a long-term research field, of a tillage field up in Willsboro, right on Lake Champlain. On the left, you can see where we've what that soil kind of looks like being held. That's Harold holding a clump of the soil. That soil in this picture has been plowed for 14 years continuously in the fall, and then we'd plant corn for grain. And we've seen the soil change over those 14 years. On the right, we've done the exact same thing in these replicated plots, except we've produced the corn without tillage. And you can kind of see, again, qualitatively, that the soil it's the exact same soil, but the different management, even visually, makes quite a difference. So if we can actually put a number on it then, and kind of move into a more using a number to discriminate what's good and what's perhaps less good in our testing. I think this is very, very demonstrative test. We will use this at field days. We have a smaller version of this that I go out to the field with and test it at right in a grower's field or at a twilight meeting. And you can see very clearly the sort of inherent structure in the soil and how that can suffer under different management or not. Okay, another test is this available water capacity test. Again, I'm showing on the right here that soil aggregate with the larger and smaller pores. I should mention that this available water capacity is defined as the difference in water content of the soil at what we call field capacity and permanent wilting point. So field capacity is that magical state that the soil reaches after about 2 days after rain, when when soil becomes in equilibrium with gravity drainage. We tend to use, you've heard of 1/3 bar. In New York, we like 1/10 bar a little better. And I should mention also that 10th bar pore size corresponds to about 30 microns, which is the diameter of the average human hair. So in a funny way, when gravity's done draining the soil, all the pores larger, if you will, than a human hair are empty now. And smaller than that are still contain water. And then as time goes on, those larger pores will, in that range, will give up their water as time goes on, with towards the end, the very small pores, the 15 bar pores, which would be 2/10 of a micron in diameter, would finally be the only ones left with any water in them. So this is a fairly standard test in the soil physics lab, and rather straightforward. And this is just sort of the underpinnings of it. Okay, soil compaction. We always want to know in the field how the soil responds. So many things, chemical test, of course, we can take the soil sample out of the field, test it, and then come back to the field with this answer and go forward. But soil hardness is something we really have to do in the field. We thought, oh, wouldn't it be nice if we could take soil cores from the field, or have growers send them in, or researchers, and then we could do soil hardness tests in the laboratory. Well, we found out pretty quick that just is too difficult. So the way we address that is to use a soil penetrometer, and you see someone using that at the bottom left side of the picture here. That's pushed smoothly into the soil from 0 all the way down to 18 inches, and we record the hardest value encountered in the 0 to 6 inch depth, and then also the hardest value obtained in the 6 to 18 inch depth. And what we're getting information on then is the 0 to 6 inch corresponds to what we call the plow layer, or the surface layer, or certainly the active layer of roots, very active. And then the subsoil is yet another environment for long-term storage of water and deeper rooting. And both of these, of course, both areas it's important to isolate these two depths. Where's my arrow? It's important to isolate these two depths to understand just where to then, if necessary, focus our management. There's some pictures you see some dense surface soil here. And the roots, again, aren't able to enter into this hard soil and take advantage of the water and nutrients that are there. And down below here, we see on the left, without subsoil compaction, the roots are able to go down and get water nutrients further down, where on the right, roots cannot penetrate that hard layer, and so are confined to growing in this little pot of soil. Excuse me. Okay, let's move into the organic, or the soil biological realm here. Keep in mind that organic matter is really the driver of all these soil biological processes. It's the organic matter, if you follow these arrows around, the first thing it does is, by having the character that it does have, and as it changes, it increases biological activity and diversity. And from this, then, there's a whole host of effects of that. I've highlighted in blue through the middle here some of these physical processes we just talked about, how when we increase this soil biological activity, we tend to get more glues that help hold these soil particles together. And so we get these nice soil crumbs, this aggregation, that are also more stable. And that said, we also then improve our pore structure. By maintaining this aggregation, we can maintain those larger, intermediate, and smaller pores so that they can all do their important functions, rather than losing or having too many of one size and not another, and then the soil can't handle those different issues. And of course, ultimately, that tilth and water storage leads us to healthy plants. But in green, we also have some important processes that are in more the biological arena. On the upper left, we see that with this increased biological activity, we tend to reduce the capacity of soil-borne diseases and parasitic organisms to function and to have an effect. So in other words, a healthy, robust population kind of minimizes the effect of these, if you will, bad guys. And of course, on the right here, as this soil biological material decomposes, it releases nutrients. It detoxifies harmful substances, and of course, we also end up generating humus and other important soil biological materials. This is a very interesting slide. This was put together by a student of Laura Drinkwater's quite a few years ago now, over 10 years. Right, so I'd like to categorize organic matter into three general types that we can then discuss. There's the living organic matter. These are the organisms and the roots. The recently dead material, which is what I would loosely call the food for the soil food web. It's the active organic matter. It's the very busy organic matter. It's forever changing. It's the stuff that's in demand. And of course, then finally, the very dead organic matter, in a broad sense, is really known as humus. It's not very reactive, but that said, it's also very important to the soil in terms of just being an important part of that soil house by loosening and holding water and just being a placeholder and a loosener in the soil. Right, and so when we talk about these living organisms in the soil, realize also that they're dying, and as they're dying, living and dying, they're providing food for other organisms in this beautiful food web. They're breaking down organic debris and releasing nutrients in a very complex, beautiful way. There's a slide here in the upper right. We show some fungi colonizing a plant root right in the rhizosphere here. Of course, we're learning more and more about this. These sort of relationships where the fungi can greatly increase the surface area of the root. And it's very useful in more perennial plants to have these beneficial fungi, but we're often at odds trying to get them to get their numbers to increase when we do a lot of tillage. And I like this picture of this earthworm on the bottom right here, especially in the context of creating and releasing glues. We talk about soil bacteria living and dying. When these bacteria die, they can each of them can burst like if you will, a water balloon, and have this glue inside them that helps hold these soil particles together. But I think also that we have to look right to the earthworm and think of the slime, if you will, that's on the skin of the earthworm. As that moves through the soil, then that slime is kind of rubbing off as well and helping bind those particles together, which we often see in their casts. So this is just again another example of this interaction of the soil biology leading to the soil to increasing that soil physical aggregation. And then, of course, the feedback of that to having an aggregated, a well-aggregated environment, this house, where the organisms can then live better. Okay, another test we do of the living organisms is the potentially mineralizable nitrogen test. This is an indicator for mineralization of soil organic nitrogen. So the organic nitrogen is first mineralized to inorganic ammonium, and then from ammonium ultimately it gets converted into nitrate for the plants. What we decided was that if we could measure the rate of conversion of organic nitrogen to ammonium, we would have a good indicator of soil biological processes. This is a very important process. A lot of organisms operate right in the same realm, and if we have good high values of nitrogen mineralization, then we'll have we have an indicator of a good soil biological environment. So this is a 7-day incubation where we look at the ammonium level in the sample at time 0, and then 7 days later. Another test we do with the living organisms is a root bioassay. Remember, this our test was developed for really vegetable crops and field crops, and so we made a choice to use this green bean. But I should mention, this is actually very interesting. The green bean variety we use is one that's very hard to find because it's sensitive to so many common diseases. But for us, that's exactly what we want. So each soil sample we receive is planted out in replicates with this bean. And so after 4 weeks, we then examine the roots of the bean and look for diseases that this bean and other vegetables are sensitive to. And then we score it according to the scale at the bottom. I should mention here that even in our current context of this test, for example, a dairy farmer is very uninterested in this test. For example, if he's growing corn for several years and then going to hay crops, he isn't interested, or it's really a moot point to him if he's got diseases in his soil that this bean would be sensitive to. Likely it doesn't anyways, because there's no food source for those organisms. But nonetheless, I think this is a good place to bring up the idea that in using this soil health testing strategy for berries or any particular plant, this is where a team of experts can put their heads together, I think, very straightforwardly, and decide, is there another plant we could use that we could do a test like this that would be more appropriate for our target crop plant. So we can talk more about that, but that's this is a very important test if you're interested in screening for disease organisms. But it should be selected. The plant should be specific towards the desired produce plant. Okay, and then now moving to the not living soil environment so much, but what we call the recently dead or available, or what we call active carbon part of this of the soil organic matter. So this is what I was calling before, that food for the soil food web. This is the material that can be literally burned for energy. So this is food, and most organisms in the presence of oxygen are using this literally plant carbon for energy, and they burn it for energy. And what this test does is use potassium permanganate to mimic that. On the bottom right, you see some tubes of purple solution. That purple solution is the potassium permanganate indicator, and as that's mixed with the soil sample, if there's a lot of material that's available to be burned as energy for the organisms, that will tend to bleach out more or less of that purple color, which we then read. Again, in the bottom left there picture, you see a little colorimeter with a cuvette that we can then read just how purple or unpurple, or how clear that solution is. So it's a very nice way it's an indicator to kind of give us an idea of, is there a lot of fuel here for the organisms or not. And then finally, the very dead humic materials. And this is, we're learning more and more about humic compounds, and they're really that black box of organic matter. It's they're kind of long-chained, complex organomolecules. They kind of resist breakdown. They stick around for a long time, and they're related to these black soils or biochar that we hear about. It's these just very complex materials that are not active in the soil food web sense, but they certainly support those sort of functions. And humus, I show a picture on the bottom left, how humus just like clay, can hold a lot of cations, which is important. Also, it acts as a sponge to hold water, which is really important on sandy soil, but clay soil as well. I mentioned before how because these humic materials are very light, it takes a clay soil which tends to get heavy and dense. It helps keep it loose and more opened. And I show some earthworm casts here on the bottom right as well that contain some very complex, as these are analyzed, some very complex molecules, slimes, if you will, but other things beyond simple slimes that really help to bind these particles together and give the soil that structure. Okay, back to our picture showing the effect of adding organic matter on these different soil processes, ultimately leading to healthy plants at the bottom. But here I've put in some red boxes, which are our soil health tests that we just went through. So the reason I like this slide so much is it shows the kind of this strategy of an ecology of an interaction. Yet what we tried to do then is target each of these processes, these functions, and with a indicator test, so we can see just how well these things are behaving, or just what sort of levels of each of these different features are in the soil. And in fact, then I put it here into a kind of a table where on the right, where's my right here, I've listed the soil processes that are associated with each of these then measured indicators. This aggregate stability, that was that soil structural test with the raindrops, where we show the soil and the sieves, and that's an indicator for our level of aeration in the field, infiltration, rooting effects, erosion potential, crusting potential, our available water capacity indicator is really giving us, again, information on the soil process of water retention. So we're going to use this to kind of understand then, when we get the results of these measured indicators, what soil processes then are good or bad or intermediate. So it's these processes are the things that interact between the different between these different physical, biological, and chemical arenas. Yet we break them out as separate indicators and processes, but we still want to, again, put them back together. And here is the product of this testing then, is our the soil health test report. Again, here's our indicators, which we just saw on the table, the physical, biological, chemical, and where the blue arrow is here, those are the values obtained. Then the next column shows a rating, where we see kind of how we then interpret the numbers. We use a red, a yellow, there we go, a red, a yellow, and a green to kind of tell us, are we low, medium, or high? And any of the values that come up in the low range, we then immediately generate these constrained soil processes. If you think why we're doing this test, we really want to know, what in the soil is not functioning up to snuff. And rather than tell a person, oh, your aggregate stability is low, what we really want to say is there's these soil processes that are impaired. And then what might you do, as I say, part two, what might you do to increase those or manage to alleviate those if we can. So I'm going to show you next where we get these ratings. And what we use is a scoring function. So what that is this is really just the normal curve, the data distribution. And if we look at just one of these curves, this clay-y line with these little triangles, we follow that from low to high. This is all the data in our database on clayey soils for active carbon. And we've split it up into the lower 30% of values in our database, the middle 40% of values in our database, and the upper 30% of values in our database. So if you have a score, if you have a clay soil and you have a score of here 550 and you're a clay, we would say, you're in the lower 30% of the database. And so we would return a red score and say your active carbon in this sample level would cause certain processes to be impaired. If we move up and that was a sandy soil and we got that same value, we would be in more intermediate range. So in other words, we see higher values in the clay than in the sand, and this is how we compensate for that by splitting the samples into different textural classes. And so again, this was developed under vegetable crops, but we believe that this sort of ranking and these sort of values are kind of idealized. Certainly vegetable crops and field crops are we're trying to grow them at a fairly rapid speed, like any plant. So these are good target values if we really want to do some discrimination. A little more interpretation here, I guess. That seems obvious, I guess. Okay, so as I said, this test has been around since 2007, and it measures, here's the 15 indicators. This entire suite of tests here is $75, keeping in mind that the chemical test at bottom here is 15, and that's included. We've also developed this basic soil health test, which takes out 2 of the rather expensive biological tests, which may not be germane to a lot of growers, particularly dairy growers. We took out the potentially mineralizable nitrogen and that root health bioassay, the root health rating. And so we were able to cut the costs and provide a product that may be more useful for landscapers or people that want to test the soil health of their lawn, for example. We're under development are ever more of these tests as we get a larger database and keep researching. We're hoping to, even in the next year, release an urban soil health test. But you'll hear more about these options from Harold Van Ness on, I think it's March 9th, 2012 in this same seminar series. Oh, and I didn't mention with the soil chemical tests here, we rank the results of this modified Morgan extraction, but New York growers also receive their the standard nutrient recommendations from Dairy One, Agro One. And this is a plug for our website, where we have all the submission forms, background info, shipping information. A lot of what you see here today is there, and our soil health assessment training manual. Just a tremendous resource. It's very colorful and easy to read, and really lays out a lot of these ideas in a very straightforward way. And that's available for free, downloadable as a PDF, even chapter by chapter. Okay, so sampling strategy. I kind of hinted on this before. We sample the top 6 inches of soil. And as we would move across let's say, call this a uniform field, you could see where we might move in this W pattern, where I have these round circles. I'm calling that my 5-gallon pail, where I put my subsample of soil. And where you see the 2 stars, that's where I'd take 2 penetrometer readings, where I'd push through from 0 to 18 inches and record at the bottom of my submission form here at 10 different spots my maximum value of the 0 to 6 inch, maximum value from 6 to 18 inches. And as I move across the field, then we get finally a sample from these 5. We composite a sample out of these 5 subsamples of soil and collect the 10 penetrometer readings. And we recommend doing this in the spring when the soil is at field capacity, or the fall when the soil is also rather damp. In the summer, we see the soil getting very hard and dry, which tends to bias our soil penetrometer readings to the point where they're really not useful. A little more about sampling. I really can't say enough about this. You'll get the results from what you put in the sample bag, um, and so I often stress with students when we talk about sampling that you have to have a question in mind that you want to answer. So let me start here with a field that has some nice representative plants growing here. Then here's a problem area. Well, what we'd recommend here is to sample this representative area and then collect that as sample 1, and then sample 2 we'd take a composite sample from this area. Then when we contrast them, we can learn just what's going on in these two different areas. Again, if we took an old-fashioned strategy, might be to take most of my subsamples from the representative area, and since my problem area is 1/10 of the field, I'll put 1/10 of the soil in my bucket from here. Well, that will give me a grand average of the field, because that if this area is 10%, but that's really not useful towards understanding and managing. And so I always like to stress that, again, that have a question in mind when you sample. Again, area 2, what's going on here? Area 1, what's going on here? We'd want 2, obviously, separate samples. But we can apply this sort of thinking to different soil types or management practices that we're trying, where plants are growing good and not so good in the field. This test, remember, it's a real shotgun approach. We get a lot of information, and we can just find out just kind of what's different between these two areas. And it's just a very educational. It's very educational to get all that information. I also want to mention this benchmark sampling, where we would sample just off the field area to kind of see, again, part of the understanding of our soil, to see just where our field stands compared to the native capacity of the soil. Okay, so here I'm going to return to this 14 years into our long-term tillage study up in northern New York on Lake Champlain. You can see the results, or you can see the results of 14 years of plowing the corn residue and planting corn again in the spring, or going no-till. What we see here is a very different looking soil. Recall, they're both Kingsbury clay loam. These are replicated plots, but you can see it's very different. Well, let's just see what's going on. And to me, this is just so telling. We would guess that we'd have some, looking at the soil, we've got some physical impediments to function compared to this soil. Well, sure enough, and we can not only confirm that, but we can put values on it, so that as we modify our management, we can track that, or we can see just how bad it is, if you will. But really, the most important thing is, I've circled in pink here these constraints. So this soil on the left is suffering from, it's got these constraints. So all these issues here are kind of listed, these processes that are in the lower 30% of the database for clays. So that when I look at my management potentials, I can try to kill multiple birds with my management stones. It's kind of the idea here. And of course, in a research setting, this is also very useful too, to get all this information to bring to bear. Interestingly here too, this steady diet of 14 years of corn stover has left us with while this is a rather good looking dark soil with good organic matter, it's kind of one-dimensional. The food value isn't very high for the organisms, and that's pointed out here. This low active carbon. So this soil we could see then that even though overall it tests high, when we look at all these indicators together versus low for this soil, we could still modify our management to try to do something to get some more food for the soil food web here. Again, this is a long-term research experiment, which we were studying tillage, but it also allows us to kind of dissect and say, what are some of these effects of tillage? Oh, and I don't think I mentioned at the bottom of these reports, it shows our the percentage of sand, silt, clay, and then the soil textural class. So the soil health testing, again, it's a nice strategy to get at the soil physical, biological, and chemical processes, giving us some comprehensive quantitative values on these individual processes. Note also that we don't give a direct recommendation from this test. We just give the values. But the real, besides education and monitoring the effects of our management, is that we want to be able to then target our management. I think this is the real goal of this testing, isn't to produce a pretty piece of paper or even to get a high score per se. It's to find out what we need to do or what we can do better. I should also mention too that certainly this low scores or red marks on this test is not an indictment on any grower's abilities. It's got nothing to do with that. It's simply that a process is below what it from our database what it could be. So that suggests then that that management to address that would be beneficial. So this is kind of puts it all together here across what we've been doing now is talking about soil processes and in the context of these soil health test indicators. Here's a grower then with this report full of all this information listing these constrained soil processes. Now he or she is wondering, what am I going to do with this information? This is just so important. This is what we mean by the holistic soil management. We're just about to move into adaptive management, but this holistic idea is these same processes that we understood to understand these test indicators. We're also going to use those processes when we think about our management, because we want to, if those processes are constrained, we need to know how management affects these processes so that we can bring them, so we can raise their values or move those values in the appropriate direction. Right, in our manual, what I show here is this is actually table 5. It gives just a broad sense table of short and long-term management approaches to deal with, where's my arrow, to deal with these different issues. So if we had a low aggregate stability in the short term in vegetables, we'd suggest adding some sort of fresh organic material. That could be a green manure, cover crop or rotation crop. It could actually be animal manure, grass clippings, who knows. But in the short term, in the long term, to deal with poor soil structure values, we might want to consider looking at our tillage, amounts of tillage, perhaps getting a surface mulch or sod crop into the rotation, etc. So this is a first cut table. Again, it's not a recommendations table per se. It's a collection of ideas and opportunities that we want to keep in mind then when we think about what ultimately we would do, what we want to help the grower come up with for what plan works for them. So these are just kind of, as I say, agronomic ideas of how things work together, and we have to have these in our mind as well. And this is, as agricultural professionals, these are just the sort of things that we can hash out with a grower to find the best result for them. And I'll show how we do that here. We've put together, this is also in the manual, this field management sheet. So it's 4 steps. It's really a winning approach to identifying problems in general. We find out what constraints, we look at the report here and see what's constrained in this field. We list, well, what might we do about those things? Again, these are all just discussion topics, but they're important steps that we want to walk through, so that we think, okay, what might I do? What might I do about aggregate stability, the short or long term? Of course, we could go straight to that table for information, but more than that, there's local experience. People attend workshops, they hear of new ideas at a trade show, online, who knows. So you kind of put those ideas here. Then step 3, we have to look at the situational opportunities, the grower we're dealing with. This person is a unique individual, their field is unique, their desires are unique. And so we need to take that into account. This person may have a lot of labor. They may be young. They may be older. May have money to invest. May not. May want to be organic or not. All this has to enter into the equation towards this goal, this final goal here, of adapting these options to the grower, the capacity and needs of the grower. So this, what we're after here is not just satisfactory, but satisfying sort of strategy for the grower. And this is it's an iterative process going back and forth, talking about what might be done singly or in combination to deal with some of these constraints, and what's available to the grower to deal with them, what they can or won't do. That's important to know. But ultimately, this is really the goal here. It's this management strategy. We really want to make this happen, and so it's that's kind of the condensate of all this testing. Right, and so for annual crops, obviously it's different than non-annual crops. If you've got semi-permanent beds here or several year beds, for example, in strawberries, this could last a couple of years. Or here's our grape example again. These grapes could be here for many years. So we're going to have different options in the broad acre versus these sort of bedded situations. So we have to keep those management options in mind. We may have more and better options here because we can bring in heavier applications and focused applications right where the plants are, and then between the plants, we don't have to dilute our application. We can focus our application, is what I'm trying to say. Keep in mind that as consultants and educators, and growers, we want to keep learning about the latest ideas, the latest technologies, the latest strategies, so that we can suggest or have a greater repertoire of management options to discuss with the grower. Because again, we have to, we start with a broad sense. It's not a recommendation, but a relationship. But we say on a case-by-case, field-by-field, grower-by-grower basis, what might we come up with for a plan? Because every field and grower combination is different, yet to be of greatest utility, I think this is the way forward. Right, and so the kind of a synopsis slide here. This adaptive management strategy is really to me the successful soil management strategy made of these main points. We want to assess our soil's health to identify constraints, and this isn't just again. I did want to say that this isn't a sales pitch for the Cornell Soil Health test. It's something that we use to develop these ideas and get them across. But it's this stepwise approach to understanding and then using that understanding to move forward with a practical, logical management approach that ultimately is the most useful. So first we want to, again, get the information, assess the soil's health. Then we want to make changes that work for that farm to address those specific constraints. Then we always recommend starting small and experiment. This is any good grower is always doing this. Trying something, borrowing equipment, renting something. He or she hears about a new cover crop, so they buy a bag of it and see how it grows on their farm. Sometimes white clover, you hear something good about white clover, and then you try to grow it and it just won't germinate. And on another field, 3 hills over, another grower, the white clover is a weed on that field and they can't get rid of it. So it's one of those things where you want to start small and just see what works. And you want to adapt these many resources of information to your farm, discussing with as many people as possible what's going on with you, learning not just about this soil science, these underpinnings of soil ecology, but you want to then know how these different processes are affected by these new technologies, be it new gadgets, new varieties, what have you. And then ultimately we're trying to build our soil so that it has an increased resiliency. The soil is the foundation. to soil vigor. And if we can reduce plant stresses with a more vigorous soil, it will benefit with increased yield and less diseases. That was my last slide. So with that, I would say thank you. And I guess if there's questions, that we can move to Kathy as moderator, and I'll try to answer those one by one.
Thanks a lot, Bob, for talking with us today. There's a question from Jeff Miller. He wants to know if there's a relationship between gross organic matter as determined by ignition and active carbon.
Great question. Yeah, you'll like this one. Yes and no. No, it turns out that organic matter loss on ignition, that's the ultimate black box. That's anything that will burn. It's I think it's 650 degrees C. You burn it, so anything that's not mineral vaporizes. But we don't know the identity of it. We don't know if it's a complex humic material, if it's fresh alfalfa meal spread on the surface, or alfalfa clippings. We don't know, and so while that's a useful number, even tracking organic matter in time, very hard. It takes a long time to get big changes in that number that you can say, oh, I'm moving in the right direction. Whereas with the active carbon, because that focuses on this particularly important part of the soil organic matter, this fuel, this delicious, nutritious, high energy material, if you will, the sugar and the candy and that quick food for organisms. That's if we have a lot of that, we can guess our soil's going to be robust and fast acting. And we can then track if we're trying to build ultimately our humic fraction, or total organic matter amount. This gives us a quicker indicator than in just a couple years we can see those levels changing and say, we are indeed moving in the right direction.
Thanks again, Bob. We'll wait and see if we have any more questions. Our next webinar is on Friday, February 3rd from 2 to 3:15. And Bob will be back with us for that webinar, and he's going to talk to us on how to improve soil biological and physical properties. If this was your first time joining us, please remember to download the pre-test and demographic survey, fill them out and send them back to me. You can send them to my email, mcm4@cornell.edu. And I want to thank Jeff for coordinating all aspects of the webinar today. He was sorry he wasn't able to join us. We're going to get him a new headset, and he'll be back hosting next week, we hope. Are there any other questions? Oh, Jeff has another one. Are there economical agents to apply to soils to increase biological activity?
Well, that sounds like the magic bullet question. Certainly there. And anytime you add material to the soil, you're going to affect the soil biological activity. And I think that's something, certainly organic growers are very aware of this. Back in the day, that's all we had. We had to consider what we feed the soil is what we'll get. And so if we want to increase biological activity, we want to increase the food. If we add a stimulant, of course, we're just going to, I don't want to say burn out our soil, but it's maybe in the short term. One thing I would say that would increase biological activity in a compost pile is nitrogen. I think everyone knows this. While that will sure break down my compost pile in a hurry, that may be good. On the other hand, if I did that out in the field, I may then have kind of burned off my by doing that adding nitrogen to a surface organic layer, for example. That will help decompose that organic matter, but it's possibly too fast, and I can't take advantage of that breakdown then. So it's one of those things where I think you have to play it situation by situation, and if you do have a soil that's loaded with organic material, but there's too much of it, and you want to get rid of it, then there are, for example, a nitrogen application and a light tillage might be just the thing to kind of stimulate that breakdown and get this release you want. But if you're trying to build that organic level, then you'd want to stay away from that and find other ways, and just try try a different approach than trying to just add some quick and dirty thing. It may be a relay crop or a time delay. I often say that one of the most powerful soil management tools we have is time, but it's something that apparently is very dear to us, and you can see why. But for example, in strawberries, often strawberries are there for 2 or maybe 3 years, but then they're removed and something else is planted there for several years. Well, it's in that then that intermediate period, that this resting stage between the next strawberry crop, that we can not only want to return some sort of income, but we want to build the soil for the strawberry, address measured or known issues, diseases, drainage, what have you, so that when we get back into the berry, we've as best we can try to take care of some of those constraints.
Well, thank you very much, Bob. I'm wondering if Jeff has another question, or we'll wait and see for another minute or two. Appreciate everyone for joining us on this snowy Friday afternoon. Okay, Jeff wants to know, many vegetable growers use their entire land base fence to fence. How would you convince them to rotate to a cover crop?
Well, I think that's just it. This is the point I was trying to make. It's one of those things you have to try it. Often it's a field day. I'm so thankful for the granting agencies, for example, New York Farm Viability Institute, that allow different growers to, they're basically giving them some funds to try things, not just for the individual that's hosting this experiment, but for others in that geographic area with a similar soil, for example, seeing how it kind of responds. And when the grower then says, "Oh my gosh, I can't believe how my soil is so different after I use this cover crop," it's kind of a I don't want to say wake-up call, but it's very demonstrative then to someone else who's wondering about how that would work. They can see it, maybe try it. They're not. It's less risky, and I think that you're going to see more and more growers finding ways to not go fence to fence with production, rather than being strung out and growing possibly lower value crops, lower quality crops, to grow a little bit less acreage of a higher value crop and building these soils that are resting. Then as these soils build, it's easier to get these better quality, higher value crops. So I think it's kind of a syndrome we're in, I think, where we're really pushing, trying to get the most out. And I think with there's lots of examples out there now where while that's a strategy, there's that may be a little bit much of a good thing. And if we could kind of merge that with some of these organic, I don't want to say resting techniques, but soil building techniques, we may find out some very useful strategies to build our soils.
Bob, would you comment quickly on your file that you uploaded for us, and then we'll close.
Oh, right. That file, I went to the Australian Wine Industry Technical Conference back in 2010. And what we talked about there was, of course, our soil health test being developed for vegetables and field crops. It's the strategy of it. It's not that you're supposed to use this test and everything will work out perfectly. That's not it. It's that it's a strategy. It's a way to get information and then use the information from this process. It's a process-driven suite of information, but then you want to manage those processes. But in a different commodity, for example, wine grapes, these measured ranges may be even nonsensical. That said, it opens up the idea for new research with experts in the field of the particular commodity. So, for example, in wine grapes, or I should say in field crops, we would say that in general, a higher value for available water capacity. So if the soil can store more water, that's better in general for field crops. But for wine grapes, that may not be the case at all. It may be even the opposite, because we want the stress of the water stress. So that's, again, not our place as to address that. That would be the work of folks in that area. Now, for wine grapes, how do we address available water capacity? And then see that the strategy's there, but then this what I call this transfer protocol. It's this. I propose to use a similar strategy. It's a good strategy, but you need to adapt it and adopt it to your particular needs. As an expert, you can say, well, no, it's that's not important for us, or it may be even a different indicator that's necessary. If you had a plant, or you're in an area that was very sensitive, very likely to have salt injury, well, of course, you'd want to have some sort of a test that would address that issue built right into your soil health testing. We didn't do that because it's not an issue with the crops we're dealing with in our test and in this area. But nonetheless, that's what this paper talks about. Is it's a strategy. It's a way to make sure you're dotting I's and crossing T's in whatever crop arena. It just makes sure that you're covering those bases so you can find out where you're constrained.
Thanks again, Bob, and everyone for participating, and we'll see you on the 3rd of February.
Great. Yep. I look forward.